Hi, my name is Biafra Ahanonu, and today I'm going to talk to you about the basic neuroscience research we're doing to understand the neural coding of pain by the brain and other regions of the nervous system. And so this is work that was done in part with Professor Mark Snitzer and Gregory Scher at Stanford University, and I'm currently a postdoctoral scholar in Alan Bassbaum's lab at the University of California in San Francisco. And so what I'm going to show you today is how we can combine imaging and computational approaches along with our understanding of pain neurobiology in order to try to address fundamental questions in awake animal sensory processing. And the hope is that this will then allow us to design and develop novel therapeutics to help alleviate pain in patients. So we're going to motivate this with a brief video. So what we're seeing here is something that commonly occurs, in which doing an activity like stretching or bending the hip can lead to pain where it normally should not. And so we'll understand the mechanisms that lead to these changes so we can help alleviate this type of pain as well as others. So pain has many etiologies, both in the peripheral nervous system as well as centrally, along with other areas that can occur. And so what we want to do is address the question of, is there a common neural code for pain and pleasantness in the brain? And how does this code change in a neuropathic state, for example, after injury? And hopefully we can use this knowledge to then develop novel therapeutics and analgesics to help alleviate pain in patients. And so we'll focus on the first two components in the basic research we're doing to understand them. So pain is characterized by a noxious stimulus that gets relayed along sensory fibers through the spinal cord as a nociceptive signal, where the brain then interprets it as pain. And this then results in protective behaviors to attempt to alleviate this pain. And so today we're going to talk about how we identified neurons in the brain that code for the unpleasant portion of pain. We will then show how this pain code is altered in the setting of injury. And then critically, we'll show that inhibiting these pain neurons actually cures the chronic pain behavior in rodents. And the hope is now in the future, we're going to try to identify the neural and molecular mechanisms that give rise to this pain code, and so that we can better understand it and develop new therapeutics to alleviate the pain. And so we're going to grapple with two main concepts throughout this talk. The first is, how does a normally non-painful stimuli, like a light touch, get transformed into a painful stimuli or percept? And for example, what are the changes in the neural activity, and what are the molecular changes that might occur? Second, we also want to look at the fact that pain has two major components. One is the sensory component of pain, and the other is the emotional or affective component of pain. So one involves what, where, and when did the stimuli occur, and the other is how unpleasant was it? And so the brain is thought to have many different areas that encode for these different components. For example, the sensory component is thought to be encoded by the somatosensory cortex that detects, for example, where the stimuli was given. And while there are many areas that involve in the emotional component, we're interested in one structure called the amygdala that has known roles in processing emotionally salient information like fearful stimuli. And so previously, what people had found is that lesions in this structure actually lead to people being able to detect that they got a noxious stimulus, but they report that they do not find it to be unpleasant. In addition to that, what we also find is that if you give patients different types of painful stimuli in neuroimaging studies, you'll see that the brain region will become activated, for example, in fMRI studies. And then also, when you look in rodents, is for example indicated here, where on the y-axis we're showing the amount of neural activity. And what you'll see is that if you go from background all the way to activating a noxious stimuli, you'll see that the neurons will become more and more activated. And what we want to know is, what are the neurons that are coding for this change, and is the similar coding in awake animals, since most prior research was done in anesthetized animals. So how are we going to do this? So in order to understand the sensory coding in awake animals, we first hypothesize that the different types of sensory inputs, like hot and cold, get coded by individual neurons here indicated in circles. And so the color of the neuron, indicated here, for example, the red neuron, indicates what type of stimuli it most likely responds to. Though we also thought that there might be some other cells, for example, these pain cells indicated in purple here, that might respond to many different types of noxious stimuli, and that they might code for the generalized aspect of pain perception. And so we wanted to know, how do these neurons relate to pain behavior? And can we measure this? So we'll be looking at this particular part of the amygdala called the basolateral amygdala, which is very involved in emotional processing and is also conserved from rodents all the way to humans, along with many other species. And so what we'll do is we'll go in and we'll use calcium imaging approaches to identify this pain code. And then we will show how we can manipulate using chemogenetic approaches to show that these pain cells are necessary for producing certain types of pain behaviors. And this is work done in collaboration with those indicated here, and I'll indicate other collaborators as I go along in the talk. And so how are we going to do this? So there are many different tools that you can use to record neural activity along with manipulate it. So one, it depends on the size of the area that you want to manipulate or record from. So for example, we're showing all the way from individual synapses in the brain, all the way to recording from the entire brain. Or you have tools that give you different timescales on which you can measure. For example, all the way from milliseconds to days, hours, and weeks, or even months. And so we are going to be using a particular approach called calcium imaging. You guys might have heard of, for example, fMRI imaging, which covers a wide range of brain areas, but you cannot record individual cells with it. On the other hand, calcium imaging allows you to record individual cells, and it can do so with a high degree of accuracy and with high temporal resolution, so it can record individual cells becoming activated in response to individual stimuli. So, how are we going to do this? So neurons fire action potentials, and these action potentials relay this information down the neuron's axon, where it will then go and activate a subsequent neuron. Now, when these neurons become activated, you can measure this by using a protein called GCAMP. Now, this protein will fluoresce when the neurons become activated due to an inrush of calcium into the cells. So what this looks like is indicated here, where on the y-axis we have the amount of neural activity, and on the x-axis we have time. And so the neuron starts off inactivated, but say we give a stimuli. And then what you'll see is once we give the stimulus, the neurons will become more activated, and then you'll see them fluoresce when this protein is expressed. And then this fluorescence will go down in time as the neurons start to become deactivated after the stimulus is done. So we can use this tool in order to understand how many different neurons respond to different types of stimuli. And critically, what we want to do is understand what goes on in awake animals. So most neuroscience studies in pain have been previously done under anesthesia, and this allows ease of recording. However, there's a change in the neural activity under anesthesia. So for example, you see here where there's very little amounts of activity in this brain region when the animal is under anesthesia. On the other hand, once you wake the animal up, there's much more and much more robust activity. So we want to understand what is the neural code in an awake animal, because then it allows us to compare this neural code to the animal's pain behavior, which you cannot do under anesthesia. So how do we do this? So if you look at the amygdala, which is indicated here, what we can do is we can express this GCAMP protein inside of the cells using a particular virus. We can then come in later with these miniaturized microscopes that were developed in our lab to record the neural activity. And we do this by implanting microendoscopic lenses into the brain and then recording the neural activity from these deep brain structures, for example, like the amygdala. And then we can go in and give different noxious or non-noxious, painful, non-noxious stimuli. And then we can go in and look before and after a chronic pain condition has set in to understand how the neural coding changes over time. So what does this look like? So if you look at the neural activity, which is going to be indicated here in the top row, and the bottom row, we're going to have the animal's behavior. And what you're going to see is you're going to see flashes of light on the top that indicate individual neurons becoming activated, while the animals initiate different types of behaviors. And so what you'll see here is this is a power of our new techniques, where we can measure activity from many different animals at once as they initiate different types of behaviors. For example, this brain region becomes activated when the animals start to initiate a movement, and so may be involved in planning different types of motor behaviors, and it is known that it does this. And so now we can start to look at, can we image neural activity in the context of pain? So again, we now have animal behavior on the top, as the animals are getting different types of noxious stimuli. And on the bottom, you have the neural activity, which is again indicated by the flashes of light. And so what we can do is measure this both in a normal state, indicated here on the left, or after the animal has undergone chronic pain after a nerve injury here on the right. And we can measure from that same animal, from those same cells, across a long period of time, shown here over a month apart. So what this allows us to do is understand how the neural activity changes across time, and critically, how it changes in the context of chronic pain. And so what we needed to do is to develop computational techniques to analyze these large-scale data sets that we were collecting. And so I'm going to briefly show you how we do that. So you have these movies where you have these flashes of light, and you need to be able to identify the individual cells and take out their neural activity in order to analyze it. So we extracted the neural activity using different computational tools. For example, CellMax is one that we had developed. It allows you to identify individual cell shapes and their cell activity. We then also developed tools to automatically clean up these automated algorithms. And then lastly, what we did, we also improved techniques to identify the same cells across time. So here you're seeing across almost 20 days, identification of the same cells. And this allows us to say, how do these cells change across time and in response to different insults? And this was developed into a tool that I made publicly available called Cheetah, named after one of our favorite big cats. So what this allows us to do is to do large-scale neural analysis. So we start off by processing the movies. So you see 4 or 5 movies here. We can then identify individual cells in those movies and extract their neural activity traces. And then we can go in and use different tools to verify that we have properly done this extraction, and then use this to do further downstream analysis. So let's now try to address the biological question of, is there a common neural code for pain that influences behavior? And how does this change in a neuropathic state? So we have our neural activity here on the left. At the same time, we record the animal's behavior from multiple different angles, and we quantitatively record it using an accelerometer. So this allows us to compare not only is the neural activity related to whether the animal does a behavior, but is the neural activity related to the magnitude of the animal's behavior, which can be used as a proxy for how unpleasant the stimulus might be considered. And so how do we analyze this? So here we have a graph in which on the y-axis, we have individual neurons, and on the x-axis, we have time. And so here where the arrow is indicated is when we give the particular stimulus, in this case, a noxious heat stimulus, and you'll see in red, there'll be an increase in the neural activity for a subset of cells. We can then look at these same cells across different types of noxious stimuli, like cold or pinprick, and you see that there's actually a common set of neurons that are activated. And we call this the nociceptive ensemble. We then go in and compare, for example, to a non-painful stimuli, like a light touch, and what you'll see is that you won't see as much activation in these cells. And we'll show later that it seems that this activation in response to noxious stimuli is specific. So what we can then do is address the question of, is this code stable across time, or is it new cells each day? And so what you'll see here is from day 1 to day 33, I can go in and identify the same cells. You'll then see that there's a subset of cells that are actually responsive both on day 1 and on day 33. Now what we wanted to do is later on, we'll go in and we'll tag the neurons on this day and turn them off on a later day to understand how they influence pain behaviors. So as a brief detour, we're going to talk about animal welfare, which I think is really important for pain research. And so what we did in order to improve animal welfare is, if you look here on this graph, on the y-axis is the neural activity. And before and after you give a noxious stimuli, for example, here and here, once you've given the stimulus, there's an increase in the neural activity, but it's not always the same each time you give a particular stimulus. However, what you can do is, using computational approaches, you can estimate, is the neuron that's activated after you give the stimulus, is it actually responsive to said stimulus? And then what we can do is we can use those models to back calculate and say, can we predict the number of stimuli that we need to give in order to determine whether a neuron is significantly responding to a given stimulus? And what that allows us to do is indicated here, where on the y-axis, we have a plot of the estimated number of stimuli that we need to give in order to be able to determine that neurons are significantly responding to that particular stimulus. And so in our studies, we use a particular level indicated here, which allowed us to trade off between getting the appropriate information to make biological discoveries while minimizing stress that animals undergo. And I think this will be important for the pain field going forward to better justify how we use animals and to better increase animal welfare going forward, while still making important discoveries that can help alleviate pain in patients. And so now that we started to show you how neural activity is related to different types of noxious stimuli, I want to show you how we can then use this to be able to predict what type of noxious stimuli was given the neural activity. So if we record neurons from the brain, what you can sometimes see is that if you say give a picture of a spider versus a picture of a tower, that you'll see one neuron will respond just to the tower, indicated by the raster plots or the individual ticks, whereas another neuron indicated here will only respond to the spider. And so what we can then do is we can plot the joint probability of the responses of any of these cells to a given presentation of a stimulus. And what you'll see is that you can make a separating line in which, in one set of joint firing, you can predict that it's most likely being given a spider, whereas in another set of firing patterns, you'll see that it's most likely that a tower was presented. And then there are cases where, if there's a joint firing, you might predict that there's a combination of a spider and a tower. And so what we can do is we can build models to allow us to look at hundreds of neurons and predict what was the stimuli given the neural activity that we see in the brain. And this will allow us to better understand what we're looking at. So what we see is here. So we can give various different types of aversive stimuli. And so we're doing this so that we can determine, are the neurons that we're seeing activated due to the fact that the stimuli is painful, or just because the stimuli is unpleasant or aversive. And so we can give unpleasant odors, air puffs, loud noises, shocks. And when we do that and we look at the neurons that are activated, here is a Venn diagram where we're showing the size of the circle indicates what type of stimuli that these cells responded to. And so what we're seeing here is, for example, half of the neurons in the amygdala that we recorded from responded to different types of aversive stimuli, and that a smaller subset, about 20%, responded to painful stimuli, but that there was still a specific subset of cells that only responded to painful stimuli. When we then go in, what we can do is we can then try to predict what type of stimuli the animal got based off of the neural activity. And so we can have some training activity, and we can use this to train a classifier that then will separate test neural activity in which it does not know what type of stimuli was given, and then we ask it to predict the stimuli. And this can allow us to then say how different are the neural activity patterns for the different stimuli, depending on how accurate the classifier can be. So what this looks like is indicated here, where you have on the bottom different types of noxious stimuli. And what they do is the classifier will predict very often that another noxious stimuli was given, but it'll make mistakes between them, which hints that there is a common neural code for those different noxious stimuli, and that it's hard to distinguish between them. Then, if you compare the activity from the aversive stimuli indicated here with the other aversive stimuli, they'll oftentimes correctly predict that a particular aversive stimuli was given, and it won't make the mistake of saying that a noxious stimuli was given, which indicate to us that there's a unique code for painful stimuli, and it's separate from other types of aversive stimuli. And we also showed it's separated from other stimuli like sucrose, which is rewarding. And so we wanted to finally ask the question, how does neural activity relate to behavior? And so here on the graph, on the y-axis, we have the escape acceleration, or how much behavior the animal initiated in response to a given stimulus. And on the x-axis, we have the amount of activation of these painful neurons. And so a light touch activates little amounts of neurons, and it also causes not that much behavior. On the other hand, if you look at a noxious stimulus, what you'll see is that it activates more of these pain cells and induces much more behavior. Interestingly, if we give a nerve injury, what we see is a somewhat of a correlate of allodynia, or an increase in sensitivity in response to the light touch. So what we see is both the neural activity increases and the pain behavior increases. And so what this led us to ask is, if we decrease the amount of activation of these nociceptive cells, will we also decrease the amount of behavior that we see, and thus have a proxy for how we can decrease pain in patients? So we did this in one way. One is there's a track in which one side's hot and one side's cold, and animals will run around until they find a Goldilocks zone. And so we can record this movement, and we can then show over time how the animal moves back and forth from the hot to the cold zones. And then what it'll do is it'll end up staying mostly in this Goldilocks zone. Then if we go in and we turn off these pain neurons using chemogenetic approaches, what you'll see is that the animal will end up spending more time in these noxious zones. And we think this is because it no longer finds them to be unpleasant, and thus it no longer has a preference towards the Goldilocks zone. Now, in order to further show what this finding entails, what we did is we started to try to separate the different components of pain. So one is the sensory, which we can look at by looking at reflexive behaviors. And when we do that, what we see is that if we turn these pain neurons off in response to a painful stimuli, like a pinprick, there's no change in the behavior. On the other hand, if we try to look at affective motivational behaviors, so running away, jumping away from the stimuli that require cognitive components of the brain, what you'll see is that when we turn these pain neurons off, you'll actually see a decrease in this type of pain behavior. And so what we think we've identified is a circuit that is involved in the unpleasant aspect of pain, yet it leaves the sensory component intact. And so what I've tried to show you today is the studies we've done to both identify a neural circuit that's involved in pain, and to show that it's involved in the sensory, or that's involved in the emotional component as opposed to the sensory component of pain. And so we identified pain neurons, and we've shown that they're related to behavior in terms of recording neural activity and relating it to behavior. We then showed that particular neurons that are responsive to light touch in the setting of chronic pain become overactivated, and this overactivation is correlated with a change in the pain behavior. And critically, if we turn off either these pain neurons or these overactivated pain neurons that respond to a non-painful stimuli in the chronic pain setting, in both cases, we can reduce the chronic pain behavior or the normal pain behavior. And more interestingly, we can reduce the behavior that's associated with the unpleasant or affective motivational component, as opposed to the sensory component, which is intact. And that can be important going forward, because for many patients, we wanted to reduce the unpleasantness, but for safety reasons, you still want them to be able to detect if they say hit their toe against the counter. And so we've identified brain circuits involved in this transformation from a normal to painful state. And what we're doing now is trying to identify what are the spinal cord circuits that are involved and how do these give rise to the circuits that we're seeing in the brain. And so one way is we're looking to see, are all the stimuli completely separated in terms of their neural coding in the spinal cord in awake animals? Or is there actually already a common neural code in the spinal cord? And that just makes it a lot easier for the brain to determine if this is just a painful stimulus or if it needs to determine what specific type of painful stimuli it is and how this relates to the behavior. We also want to know the changes that you see, for example, in the spinal cord, paralleling changes that we saw within the brain. And so one of the things we want to do is, beyond just looking at the spinal cord, we want to understand how do downstream brain regions use information from these pain neurons in order to initiate the proper behaviors given the context. And so, as we talked about before, there's stimuli coming in from the periphery, and it goes to the spinal cord, where these projection neurons send it to the brain. And there's many brain regions involved, as we talked about. So we talked about this amygdala and its role in producing pain behaviors. And so what we're interested now in is these downstream areas that receive outputs from the amygdala. So for example, the frontal cortex, which can be involved in emotional regulation and determining the proper behaviors to initiate given the context or what is going on in the world at the time. Or other structures like the striatum, which might be involved in movement and determining what specific behaviors to initiate and how to initiate them. And so to record from all these different regions, or to better understand how multiple brain regions coordinate their activities to produce these complex behaviors in response to pain, what we're doing is we're looking at many different areas. So we talked about the emotional and the sensory component, but there's also a motivational as well as a motor component of pain and a visual component in terms of anticipating painful stimuli. And so what we can do is we can record from the entire cortex of an animal to determine how different brain regions interact in order to produce the proper pain behavior and pain percepts. So to do this, we had developed a technique called the crystal skull, which was originally developed by Tony Kim and Yangping Zhang in our lab at Stanford. What this allows us to do is go in and replace the entire portion of the mouse's skull with glass, and then allow us to go through and image the neural activity from these large areas. What we can then do is indicated here. We are seeing many different areas within the mouse cortex, and all those individual flashes of light are individual neurons that are becoming activated. What we can then do is use the computational approaches that I developed while a grad student to be able to identify over 10,000 individual neurons across multiple different brain regions. We can then go in and overlay the known brain regions on top of the cells that we've identified. And what this will allow us to do is now, for the first time, be able to understand how many different brain regions coordinate their activity to both sense incoming stimuli, determine the context for that stimuli, determine where it's coming from, and to determine what type of behaviors to initiate based off of the animal's motivational state. For example, if it's happy or if it's sad or depressed. And I think this will allow us to better understand and identify novel targets for pain therapies, along with just understanding the basic mechanisms of pain. And so I've shown you today how we've tried to identify the neural properties that lead to the coding of pain in the brain. We are now trying to identify the molecular properties of these pain responsive neurons. And the hope here is that we can then develop new therapies. In part, this will allow us to look at individual cells indicated here, where we can then go in and say they may have changed in a chronic pain setting. And we can then go in and inhibit a specific subset of those cells indicated here, or inhibit a particular protein to reduce the neural activity to hopefully alleviate pain. And so we can target different parts of the pain pathway using these novel therapies to then eliminate pain in patients. So I've shown you how we can go from an abstract view of pain to a mechanistic understanding. This will allow us to both simulate pain, which could potentially allow people to better understand other people's pain. We also want to understand the psychosocial environment so that you can understand, for example, how pollutants or other aspects of the environment can affect the development of chronic pain. And lastly, we want to use all this knowledge to be able to finally eliminate pain in chronic pain patients. And so I'd like to thank all those in the SEETS lab, especially those indicated here, who helped with the first part of these studies. And I'd like to thank all those in the BASBOM lab who have started to help me better understand the pain processing within the spinal cord. And so everyone that's been involved has been a great help, especially Jones Parker, as well as Gregory Corder, both for their mentorship while I was a grad student. And so I look forward to taking any questions on the many tools as well as biological findings related to pain that I've shown you. And I look forward to discussing with people at the San Diego Pain Summit in February. And I hope everyone has a great rest of the day and a great rest of the conference.